You've probably heard of cloning, making identical copies of things, including reproductive cloning of organisms. But what is gene cloning? Making multiple copies of a specific gene is gene cloning. Bacteria are relatively easy to grow in the laboratory, so what if we could genetically manipulate bacteria to produce valuable proteins for us to use? For example, we could get bacteria to copy the gene for human growth hormone and then make large quantities of that gene product, which we can use in treating children who need it. That's exactly what we can accomplish through gene cloning. The general idea is to make recombinant DNA by joining a human gene to bacterial DNA. In this case, we start with bacterial plasmids, small loops of DNA outside the regular chromosome that are readily replicated and can serve as gene carriers or vectors. We then let bacteria take up the recombinant plasmids, grow, and reproduce. They make copies of the human gene and express that inserted gene as if it were their own. We then can collect the protein they produce and use it. The first challenge in gene cloning is making recombinant DNA. To do this, we need enzymes called nucleases to cut the DNA, and the enzyme DNA ligase to join the pieces together. Since we want to control exactly where the DNA cutting occurs, we use restriction endonucleases. These hydrolytic enzymes digest phosphodiester bonds only at specific DNA sequences. Bacteria normally produce these enzymes in defense against invading viruses, but we are going to use them in the lab to make recombinant DNA. Each restriction endonuclease recognizes a specific sequence of bases and only cuts DNA at that site. The short restriction site is said to be palindromic, meaning that the sequence on one strand of the DNA is exactly the same in the opposite direction on the complementary part of that DNA. 3 enzymes and their specific restriction sites are shown here. Many useful restriction enzymes make a staggered cut at their restriction recognition site. So, after hydrolysis, both of the pieces will end in a so-called sticky end. If those pieces realign, complementary base pairing can again occur. To prepare recombinant DNA, the 2 sources of DNA are cut with the same restriction enzyme, producing complementary sticky ends. When the fragments are mixed together, the sticky ends form hydrogen bonds between complementary base pairs. Through this process, 2 DNA molecules, originally from different sources, can join together to form a new molecule. DNA ligase can now seal the phosphodiester linkage, and we have recombinant DNA. Once we have our recombinant plasmid, we make the bacteria take up the new DNA through the process of transformation. However, a big challenge in gene cloning is to know whether or not our bacteria cells contain the recombinant plasmid. Some bacteria don't take up plasmids readily. Even if they do have plasmids, how do we know whether they took up the recombinant plasmid with the inserted gene or one of the original plasmids without the human gene? We obviously cannot ask the cells individually, so we design the plasmids ahead of time to contain marker genes. We can construct plasmids that contain genes for antibiotic resistance. In this example, if the bacteria are able to grow in a medium containing the antibiotic ampicillin, we have evidence that they have taken up the plasmid with this marker gene. We then select only bacteria that have the plasmid to grow in large quantities. We can also make plasmids with genes that produce a colony color change, as in this LAC-Z preparation. If the gene to be cloned is inserted in the middle of such a marker gene at one of the restriction sites shown, then the color change does not occur. This means we can screen for bacteria that contain an inserted gene. In this example of gene cloning, we are asking a prokaryotic cell to express a eukaryotic gene. You already know some of the differences between prokaryotic and eukaryotic genomes and gene expression, so consider how this lab process of gene cloning must be tailored to bacteria. For example, When inserting the human growth hormone gene, can we use the human promoter? No, bacterial RNA polymerases will only recognize a prokaryotic promoter. Furthermore, if we insert a full human gene, the resulting polypeptide will contain a set of correct amino acids, then several amino acids that don't belong in the protein at all, then another normal part of the polypeptide, then more amino acids that are garbage, and so forth. The protein is much longer than normal and contains intervening segments that don't belong. What's going on here? Remember that human genes have introns, and bacteria cannot splice out introns. So we can't use the human growth hormone gene straight from human cells. Instead, we have to use a cDNA version of the gene that contains no introns. This cDNA, or complementary DNA, is made in vitro in the laboratory from messenger RNA that has already been spliced and contains only exons. This process, making DNA from RNA, is not typical of either prokaryotic or eukaryotic cells. So in the lab, we use a retroviral enzyme called reverse transcriptase to create the cDNA. Now, if this cDNA is taken up with the plasmid, the bacteria can translate the normal human polypeptide without the intervening introns. There's still another issue with bacteria producing human hormones. In our cells, human growth hormone is packaged up for secretion by the Golgi apparatus. But bacteria do not have this system of membranous organelles, which is required to chemically process and package the human gene products. To resolve these issues, we can use gene expression systems in simple eukaryotes like yeast to get our gene products, or we can find other ways of extracting proteins from the bacterial cells. Using bacterial plasmids for gene cloning has become an extremely successful approach for the production of diverse proteins in industry. Biotechnology companies have scaled up the production of purified hormones and enzymes that are important in medicine and research. Therefore, from understanding basic gene expression systems in microbes, we are now able to solve many applied problems as well.